Now let's discuss what each of the specific components of the cellular membrane are going to do for the cell membrane as a whole. Each of these things as a constituent unit, so for example the phospholipids, the proteins, the cholesterol, the channel proteins, all of those things play a specific role to bring about the proper functioning of the cell membrane. So let's look at what each of these things do. So before we actually begin, I'll give you a small list almost of the things that we will be discussing. First of all, we have to talk about what the phospholipids do. Then we'll be taking a look at cholesterol. It's very important. I know cholesterol is a very small molecule, if observed here, but it plays a very important role. Then I can teach you about the glycolipids and proteins. Glycolipids and glycoprotein. And then we'll talk about the proteins, the specific proteins as a whole. Let me align all of that like this. Okay, better. Let's begin with phospholipids. I'll use green. So number one, phospholipids. I should increase the size of the marker. Phospholipids. Right. If I draw a phospholipid, you should know the structure of a phospholipid already. It's very simple, like this. We have a head, not just a head, it's going to be a hydrophilic head, meaning it likes water. We discussed all of this under biological molecules, and you remember the tails are hydrophobic. Hydrophobic tails. I will type out all the functions. Now let's actually write the first statement as a thing. Have hydrophilic heads and hydrophobic tails. Point number one. Now here's the deal. Phospholipids makes the intermembrane space hydrophobic. Did I spell space wrong? Space hydrophobic. What does that mean? Now scrolling up, this is what it means. Here, this is the intermembrane space. The intermembrane space means roughly the space in between the two membranes or the two bilayers. We're talking about roughly this region. Now you see all tails, no? All tails meaning what we're going to have is an overall hydrophobic nature. So it makes the intermembrane space hydrophobic, therefore prevents water-soluble molecules from entering or leaving the cell. Now, what does that mean? If I take a good look at this membrane, and I'm going to introduce a water-soluble thing. I'll draw it in blue. Right. What does water-soluble mean? Water-loving. Or in other terms, in most scientific biological nomenclature, hydrophilic. That's what water loving means. So that means it has no problem. This guy has no problem to interact with the phospholipid heads because they are hydrophilic themselves. But they cannot cross through this space into here because once the molecule reaches the tails, what is it? It's a hugely hydrophobic nature. It's going to have to counteract. It can't do that. So there's no way that this molecule goes through this like this. That's not possible. Okay, that is why water soluble things need special channel proteins. Okay, that is actually a very counterintuitive thing when it comes to the study of cellular membranes and the biology. You might think that water soluble things have no problem. Water soluble things have no problem at all. They can just cross through. Wrong. It's the opposite. Water insoluble things, meaning hydrophobic things, they can cross through without an issue. But if you are water soluble like this guy here, you're going to have to take the route of a channel protein or a carrier protein or something like that. Okay, sad news. So water soluble molecules cannot just enter, cannot just leave the cell. Okay, now phospholipids are capable of forming two types of structures. So this forms two distinct structures when put in water. What does that mean? Let's see. Now, if I were to take some phospholipids and put them in a jar of water, try my best to draw a jar of water, and now take the phospholipids and I'll draw them in pink. Okay, if I take a couple of phospholipids, let me replicate them. Okay, 2, 3, 4, 2, 3, 4, 2, 3, 4. Right. Now, if I just put them in water, they're not just going to stay like this. Okay, they won't just stay like this. That's not going to happen. These phospholipids have hydrophilic heads and hydrophobic tails. So they will orient themselves accordingly. Now this is full with water now. So the hydrophilic heads will turn themselves towards water. But once you get to the hydrophobic tails, they don't like water. They try to hide as much as possible. So something like this happens. I'll draw all of it. So they will orient themselves like this. They are going to orient themselves like this. I hope you see where I'm going. Uh-huh, 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 like this. So continuously, if this happens, all the heads will face outward and all the tails will be inside. Okay, it's a circle with the hydrophilic heads facing the water. They have no problem. They're hydrophilic. They love water, so they will be facing the water. But the tails, they don't like water. They're hiding. They are hiding. We call this structure a micelle. Micelle, or sometimes myceli. It's silly to call it that, but something like this. And you know the shape, it's spherical, so we call it a spherical mycele. Mycele, micelle, myceli, it's fine as long as you can write it properly. Okay, that's the first structure it will form. And it can also form this structure. What is this structure? What have we been discussing about so far? We have been talking and we have been discussing about bilayers. So they will orient themselves like this. So all the tails, like so. And now you have this. So this is simply a bilayer. Two layers. What's the important property to understand here? Tails will hide from the water, heads will face the water without any issue. Without any issue. Now, if you just take one part of these things with the tails are missing, Lord, you can even call it that they can form a monolayer. Monolayer. Okay, but the most important structure that they're going to form are the mycele and the bilayer. Okay, that is about phospholipids. Now let's move on to cholesterol. This is going to be an interesting one. Phospholipids. Number two, cholesterol. I drew cholesterol just like this amongst the bilayer, and this is going to play a crucial, very important role in the fluidity of the membrane. What am I talking about? The fluidity of the membrane. So bullet point 1, I will say cholesterol's main purpose is to regulate the fluidity of the membrane. You can think of the fluidity of the membrane like the, how am I going to put it? The strength of the membrane. No, it's not really validated as strength, but you can think of it like the integrity of the membrane, like how hard the membrane is. Okay, how hard the membrane, you'll get that memo, I swear. Now let's move on to the next point. It's almost like the strength of this thing. So I'll just draw a happy face here because we couldn't come to a conclusion. Right. Cholesterol can give you an increased mechanical strength to the membrane, increased mechanical strength, and stability of the membrane. And stability of the membrane. Let's look at what happens under specific temperature. Let's see how this thing actually regulates the fluidity. Now, first of all, I'll discuss about the scenario that happens at low temperatures. Reduce the pen size. Okay, that's better. At low temperatures, cholesterol will sit between the phospholipids. Between the phospholipids. And by doing this, they will preventing them from packing too closely together. Okay, let's put this into a little bit more of a understandable notion. Okay, look at this. So I have a couple of phospholipids. Right, I'll just use one layer because that's going to help you understand better. Now, if these phospholipids pack together too much, see, if they're packed together too much, like this, there's a very good chance, there's a very high chance that the membrane itself collapses, that the membrane itself will break. Nobody wants that. We don't want the membrane to break. Okay, we want it to maintain a threshold fluidity. What do you mean by a threshold fluidity? That's something I came up with right now. Simply speaking, not too hard, not too soft. Okay, so at low temperatures, what's usually going to happen? So temperature is low. Kinetic energy is very low. Temperature is low, kinetic energy is very low, so everything tends to pack together too closely. Cholesterol in this case prevents that. So for example, if you now imagine if you have some cholesterol molecules amongst the phospholipids, this is going to prevent them from packing too much together. It will leave some space in between. So at lower temperatures, the cholesterol will prevent the phospholipids from packing too closely together. Too closely together. Okay. Now, there's actually more factors that go into the fluidity of the membrane. So let's discuss those one by one. The fluidity of the membrane is affected by semicolon. Point one. Unsaturated fatty acids. We're talking about the fluidity of the membrane, alright? The fluidity under cholesterol. The main factor we have to assess, the main factor we have to talk about is the fluidity of the membrane. Maintenance of the fluidity of the membrane. Right, first of all, unsaturated fatty acids. unsaturated fatty acids. What do? Unsaturated fatty acids make it more fluid. So I'll draw an up arrow to indicate increased fluidity. But how and what's the notion? It's like this. If you have the phospholipid, the tail is a fatty acid, obviously. So these tails are fatty acid tails. Now I'm going to zoom in to get a better idea. If you have a saturated fatty acid tail, it's straight, right? If it's a saturated fatty acid tail, it's a pretty straight tail. See, like this. Because all the carbon-carbon bonds, why did I put a Z there? Sorry about that. Leave Z, okay. All the carbon-carbon bonds are single bonds, so it's going to be a straight chain. You learned saturation and unsaturation in IGCSEs and IGCSEs and O levels and organic chemistry like this. Now, the moment a double bond is introduced, the chain becomes unsaturated. That we know. If we have a double bond, that means unsaturation. So the thing is, the moment a double bond is introduced, the chain will have a slight change in length like this. It was pretty straightforward before, but it has a small kink, a small bend to it. A small bend to it now. So now what happens to the packing of the phospholipids? I will show you two scenarios. Honestly speaking, you don't have to learn this part this extensively, but I always feel like teaching it to this extent helps you understand better. So yes, you will learn to this extent. Right. So these are all saturated fatty acids. See, they can pack very closely together. They can pack very closely. The tails can come very closely together. So we can say that this scenario here is less fluid. This is less fluid. Okay, fluidity is down. Now I'm gonna try and pack this. Do you see? Now I can't go this close because the fatty acid tails are gonna touch. So I have to at least leave this much of a space. At least. Let's get rid of him. Technical difficulties. Okay, right. See? Now look at both scenarios. See? Do you see now that this is much more fluid? It's much less packed together. It can move better. There's more space between the phospholipids. But here, this is packed together. That's the idea of saturation and unsaturation. If you have unsaturated fatty acid tails of the phospholipids, more fluidity. It's a very simple scenario. I'll move those structures here. Like so. Okay, point 2. The next thing that I'm going to tell you is the opposite version of that. Saturated fatty acid tails make it less fluid. Now you know. So fluidity down. What about temperature? It's a very simple scenario. I should use red for temperature. What does a low temperature mean? Less kinetic energy, less movement, everything tries to pack together. So obviously, low temperature means less fluid. Okay? And at the same time, high temperature means high temperatures make it more fluid. Everything's moving. Everything has more kinetic energy now. So everything's moving, more fluid. And cholesterol regulates that. Having cholesterol will increase the fluidity. No cholesterol will decrease the fluidity. That depends on this said scenario. So I'll just leave cholesterol as a factor here like this. Okay, the last factor, now the first factor that we had to talk about was saturation, unsaturation. Remember, these are factors affecting the fluidity of the membrane, okay? You will have MCQ questions talking about the specifics of these fluidity dependent factors, for example, saturation, unsaturation, temperature, presence or absence of cholesterol in that sense. So first factor, saturation, unsaturation. The second thing that can affect membrane fluidity is the temperature. The third factor would be the presence or absence of cholesterol. And as the fourth and final factor, what we have is the tail length, the phospholipid tail length. This is also a very obvious and very straightforward. If you have longer tails, zoom in for a better picture. If you have longer tails, of course, you're going to have more attraction between the tails because tail length is longer, more bonds. Now, let's compare these to very short tails. Okay, see, just 2 bonds, two intermolecular interactions. But here, there's a lot. If the tails extended even more, you can have even more bonds. See, more space for more bonds. What does this mean then? More bonds, more interactions, what will that result in? That's going to result in higher intermolecular forces. They are going to be closer together. That means they're going to be too packed together. So less fluid. So less fluid. You'll understand this when I throw this into a concluding statement right here. Longer the tails, less fluid. Why? Why is it less fluid? Because more interactions between the tails. So the phospholipids, each of these phospholipids are going to have a hard time moving around. They're going to have a hard time moving around. That's the case. All right, that is the case. Let's write the opposite scenario of this. Shorter the tails, more fluid. So up arrow, more fluid. That is the fact, those are all the factors that we have to discuss about cholesterol. And why are we talking about the fluidity of the membrane? Because cholesterol is one of those factors that determine the fluidity, the fluidity of the membrane right here. Okay. Now let's talk about the third factor we have to talk about, the glycolipids and the glycoproteins. Let me reset zoom level. It's 110. Going down. Glycolipids and glycoproteins. This is 3, right? 1, 2, 3. Glycolipids slash glycoproteins. First of all, let's understand the morphology. Here's a lipid, a phospholipid. I'm connecting a small glucose or a carbohydrate piece. I have a glycolipid. Here's a protein. I'm connecting another small this thing. Now I have a glycoprotein. Glyco, that's the lipid part. Glyco, that's the protein part. Okay. Let's type for this. Now both of these things, they contain, let's spell carbohydrate, carbohydrate chains that exist extrinsically. What does that mean? Carbohydrate chains that exist extrinsically means, do you see? I go to this. This is the end of the membrane. So this is the inside of the cell over here. And that's the outside. So that's what you mean. These are extrinsic. Now there's a very good reason why these are extrinsic, and there's a very good reason why everything is pointing outwards. That is to help cells to communicate with each other. That's to help cells signal each other. Okay. We'll talk about cell signaling after this. That's unit 4.3. We're still under 4.2. Understanding the morphology of the cellular membrane. So both contain carbohydrate chains that exist extrinsically, which enable them to act as receptor molecules. Receptor molecules for what? For example, we can have signaling receptors. Then we can have receptors involved in endocytosis. What do you mean by endocytosis? It's a form of transport. More about transport later. And receptors involved in cell stabilization. You don't want to think about that too much. Okay, and they can act as antigens for cell-to-cell recognition. If you have no earthly idea what antigens mean, I recommend you don't go and Google that. It also is signaling spelled with one L. Oh, interesting. Okay. I think this is the mistake here. Stabilization. Yes. Don't search what antigens are, but just know using these glycoproteins, using these glycolipids, cells can communicate with each other. That's what you mean by cell-to-cell recognition. Cells can communicate with each other. Final part, let's discuss what proteins do. Fourth and final part for 4.2, the proteins. Now the proteins embedded within the cell membrane, if they are inside the cell membrane, you call it intrinsic proteins. It is an intrinsic protein called intrinsic proteins. Okay. A similar name for intrinsic proteins you will sometimes see in question papers would be integral proteins. Same idea. If they span the entire membrane, if they span the entire membrane, what does that mean? Let's put that into perspective. Going back to the cell we drew. See, this thing spans the entire membrane. This thing also spans the entire membrane. This membrane and this membrane. Does this thing span the entire membrane? Clearly not. If they do span the entire membrane, you call it a transmembrane protein. We're just talking about some nomenclature, how things are named. It's a transmembrane protein. Typically, almost all the transmembrane proteins are actually transport proteins. Almost all of them, not all of them, okay? Almost all of them are transport proteins. So on the contradicting statement to the first one, I can say something like this. Proteins are also found in the inner or outer surface of the membrane, inner or outer surface of the membrane. These are known as extrinsic or extrinsic or peripheral proteins. Again, to clarify the statement, I shall go up and show you the extrinsic protein. Yeah, this guy. He's just outside. He's not spanning the cell membrane. He's just outside. Extrinsic or integral protein. Sorry, extrinsic or peripheral protein. So when you're taking down the note, make sure you mark which thing each point is referring to. It's going to help you learn better. And like I've said before 8,000 times, when it comes to biology, diagrams are going to be your best friend, talking from experience. Right? Okay, now one last point here that I would like to mention. Now I told you that if a protein spans the entire membrane, we call it a transmembrane protein. Okay. And most of the time, these transmembrane proteins are almost all the time transport proteins. Transport proteins can actually come in two types. I think I should mention the two types for you. The first thing is we have a certain set of proteins known as channel proteins. And then we also have another type of, or another set of proteins that help transport, known as carrier proteins. With that, that's the end of the things, the pieces, the components that are going to make up the cellular membrane.